Jason Haugh is one of our longtime friends. He's been helping us as an advisor for our center grant for, I don't know, 15 years maybe. He's a world-renowned cell biophysicist, has done both experiments and modeling. He's super well known in terms of his modeling of reaction systems associated with membrane receptors. And I think he'll tell us a little bit about that today. And we're very grateful, Jason, for you accepting this invitation to be our first plenary speaker.
All right. Thank you, Les, and thank you for the invitation. And I'm happy to talk about VCell. I want to make sure that you're going to see my screen properly here.
So far, so good. Yep.
Okay. Are you seeing the presenter view, or are you seeing
presenter view?
Oh, okay. Let me switch that.
No. You were seeing the slides. We're seeing the slides.
Sorry, we're seeing the slides. Yeah. I meant, yeah, we're okay. Seeing your presentation.
Okay. Are you still seeing it?
Yes.
Okay, great. Yeah, I'm happy to be here virtually. I first came to learn about VCell back in 1999. So in the previous century. I mean, VCell is almost, as far as I know, 30 years old. And it took me a few years to come to know about it. During my postdoc, I overlapped with Chuck Fink, who former alum of the program there, and he used VCell in his PhD work, and so that's kind of how I came to know about it. I met Jim Schaaf at a conference maybe that year or the next year as VCell was becoming popularized in the community. And then, as I'll explain, I finally made the decision to start using virtual cell in about 2005, and I'll explain why that's the case. I guess my initial hesitance is because, as you can see, I'm a chemical engineer by training, and we learn about modeling and sort of taught to use. I think, well, this will sort of show my age. I learned Fortran. The ethic was that you coded everything yourself, and if you were using
No, cześć.
I'm sorry, could you please mute yourself? Thanks. Right, so there was this ethic of coding everything yourself and hesitance to use some kind of off-the-shelf package. So I'll explain why I made that, it changed my mind about that. Okay, so first some background on signal transduction, and I'll go through this pretty quickly. Basically, signal transduction networks are the biochemical pathways by which stimuli in a cell's environment are decoded, if you will, as instructions to basically engage in some kind of behavior or function, right? So, rather than say, tracking carbon or energy like you would in a metabolic pathway here, I suppose the currency is information. So the cell is going to perceive these stimuli and basically decide, to change its state or function. So I listed here different cell states and fates: quiescence, proliferation, autophagy, differentiation, apoptosis, and senescence, and cell migration, which I'm going to talk quite a bit about. And of course, when we talk about signal transduction, we can think about the normal function of cells in our body and in process such as like development, immunity, wound healing, and angiogenesis, and how different cell types have to communicate with one another in those processes. And of course, when signal transduction goes awry, this is the basis for pretty much all of the important human diseases: cancer, immune system disorders, metabolic disorders, and others. And so an example of where a signal transduction network can go awry is if you have a non-functional network, that would be a situation where the stimulus is provided, but the cell doesn't respond to it, right? Because there's some kind of one of the proteins that's involved in this network is not functioning properly. And in the other case, an unregulated network is where the stimuli are not being provided, but the cell is sort of doing its own thing and responding autonomously. And both of these types of dysregulations are very important in cancer. And I like to say that if you're doing signal transduction research, you are inherently doing cancer research, because any number of important pathways can be sort of wrecked or caused to be autonomous in cancer. So this is now, as you can see, a very old diagram showing some of the signal transduction pathways in the cell, and the ones that are marked in red here indicate that they're prevalent in human cancers, right? So one protein that a lot of people like to point to is RAS, which is a small GTPase, and it's mutated in somewhere between 25% to 35% of human cancers, depending on which database you believe. And so we can think about pathways that are involved in cell growth, proliferation, survival, and cell migration, right? So cancer cells, they grow uncontrollably, they proliferate uncontrollably, they ignore growth inhibitory signals, they ignore cell death signals, and some cancers become invasive and migrate from the primary tumor so as to form other tumors colonized in other places in the body. Of course, at that point, the prognosis of the patient is very dire. Now, if we want to try to model this process, signal transduction, it's pretty difficult because there's a lot of different pathways that one can look at, and sort of the overall, I guess, thing I would say is that integration from the molecular level, connecting the dots in the network, to a systems level, how do all these pathways work together and to affect the cell's behavior? We're nowhere close to that level of integration, right? And part of the issue is that drilling down to the mechanistic detail is very hard. Any arrow that you might see in a reaction network, was likely several PhD theses or postdocs who worked on that very specific process. So what we're trying to do is try to build this up from a multi-scale point of view. And when I say multi-scale, what I'm really talking about are different scales of biological abstraction, right? You could go from the very finest detail, thinking about proteins at an atomic level, right, in terms of their structure function, right, and then how those proteins interact with one another to form complexes. That would be yet another scale, and then extrapolating to networks and the whole cell, and then finally even to tissues in an organism. And so our approach is to do experiments and modeling mainly at the level of the individual molecules and pathways and in individual cells, and then sort of extrapolate either from the structure function side on the one hand and the cell tissue interface on the other side. Signal transduction networks have an organization to them. At least that's the way I like to think about it. Most stimuli are sensed by cell surface receptors, and then once activated, those receptors will recruit other proteins to the plasma membrane of the cell. Some of those proteins are considered adapter molecules because their function is basically to mediate recruitment of other things, right? But they don't have catalytic activity. They're not enzymes. And then other, shown here in blue, this next layer are enzymes that are recruited either directly to the receptor or via one of these adapter proteins. Once recruited to the plasma membrane, generally these enzymes act on substrates that are also localized in the plasma membrane. So these are lipids or lipid tethered proteins. And the protein I mentioned before, RAS, is an example of one of those lipid tethered proteins. And so recruitment to the plasma membrane is integral to sort of the function of these enzymes, because you're bringing the enzymes in close proximity to its substrates. And so already you can see that there's a compartmentalization aspect of signaling that plays really nicely with the kind of modeling that you can do in VCell. And then so these are all lipids or lipid tethered proteins, and then they in turn recruit effector enzymes. These are all serine threonine kinases, actually, also to be activated at the membrane. And then from there, you can have transduction of the signaling through the cytoplasm and also in the nucleus of the cell. Okay. So most of what I've shown you here is very proximal to the receptor and the plasma membrane. You can also see that within this network, there's a lot of so-called crosstalk between individual pathways and also feedback, right? So it gets complicated very quickly. And it's not just a simple, sort of linear sort of flow of information. All right, so getting into some of the work that we've done, I'll just give a couple of quick examples before getting into the main part. So this was some work dealing with a pathway called the ERK pathway. RAS is an important protein in that pathway. If I just step back for a second here, that would be pretty much this left side of the network here. And here's our friend RAS. And then you can see that there's 3 kinases that get activated, ending in the activation of ERK, which is a so-called MAP kinase, mitogen activated protein kinase. All right, and one of the things that we observe, just like anyone who's done measurements on this pathway just about, is that there's adaptation. If you were to measure the phosphorylation of ERK as a hallmark of its activation, it's transient, right? And that's that you can see sort of that response here, right? But when we looked at activity using FRET biosensors in individual cells, either targeted to the cytoplasm or into the nucleus, we didn't see that adaptation, at least not to the same level, right? And so this begged for some kind of kinetic model to try to reconcile that. And we did. We found an explanation. And that explanation then can lead to further predictions. Right? The explanation that we found was also hinted at in the literature, and that is that ERK interacts with many substrates. It has well over 200 putative substrates, which are divided almost equally between the cytoplasm and the nucleus. And the interactions of ERK with all of these substrates was sort of buffering it. And then as the ERK sort of phosphorylates those substrates, then that shifts sort of the steady state, if you will. And so that was a pretty satisfying explanation. And we did sort of a lot of model fitting. So we have the kinetic model, we fit it globally to this complex data set, right? That was the approach. This is the work of Shoaib Ahmed, former student. And then another example, we're looking at the AKT-mTOR pathway, which is very important in the controlling metabolism, right? It's sort of this pathway switches on protein synthesis and also regulates autophagy and apoptosis. So also very important for cell physiology and also very prominent in cancer when this pathway is awry. And here again, we're taking sort of these measurements and fitting them to models and then trying to understand. We make model predictions and test them and refine the model if we need to. And then we try to make some sense out of it. We do sort of a post-analysis of the model and say, okay, what is it about the model that made it fit a certain way, and what can we learn from that? Right? This was the work of Ahmed, Rahman, another former student. Now there's a problem, or I guess an elephant in the room here, and that is I already said that space is important, or at least compartmentalization is important. But all of these kinetic models are just ordinary differential equations in time. And most of the measurements that were made here are what you might call population endpoint, right? Where you take a population of cells and then you make a lysate, and then you probe that lysate biochemically, right? So the type of modeling sort of matched the type of data that we had, right? But there's several issues with that grind and find approach, the population endpoint measurements. One is that you're taking it over a population of cells rather than individual cells. And so that means that you may have, basically each cell doing its own thing with respect to time, and then that sort of gets averaged in some way that's not representative. That's a possibility, right? More importantly, I would say, is that all spatial context is lost, right? When you dissolve cells, right, you can't tell where the molecules were prior to that, right? So in parallel with this kind of research, we're also doing live cell microscopy, right, where we're looking at individual cells doing their thing. They're expressing some kind of fluorescent protein that we're looking at. Either we're tagging a protein that's normally in the cell, or we're tagging a domain of a protein that will bind to some target and give us a readout of that target, right? So a couple different approaches there, right? And what we're measuring is either the translocation of that fluorescent protein, say from the cytosol to the membrane, or from the cytosol into the nucleus, or we have some kind of biosensor that will change its properties when it changes its conformation, or when it's bound to something in the cell, right? So you have these so-called FRET probes, or solvatochromic biosensors. Anyway, so we can hopefully be able to measure in an individual cell what's going on. And so we did a lot of what's called TIRF microscopy. That stands for total internal reflection fluorescence microscopy. And what happens in TIRF is you basically will come in at this very shallow angle here, such that the beam is totally internally reflected, but then you produce an evanescent wave that excites the cell above, but only penetrates about 100 nanometers. So for a mammalian cell, that's only sort of the footprint of the cell. So you're basically capturing only about 5% of the volume of the cytoplasm. But it's very good for looking at things at or near the plasma membrane, at least the plasma membrane that's in contact with the surface, the contact area. And so in the movie that I'm about to show you, the cell is expressing GFP, green fluorescent protein, tagged to the plexurin homology domain of AKT. And this AKT pH domain binds specifically to particular lipids that are present in the plasma membrane that we were studying. And so the more abundant this lipid is in the membrane, the more our GFP AKT pH translocates to the membrane, and hence the higher the intensity as viewed by TIRF. Okay, And so in the biological context is this lipid PIP3, which is produced by an enzyme called phosphoinositide 3 kinase. That is upstream of that AKT mTOR pathway that I mentioned already. But in this context, what we were interested in is its function in stimulating F-actin polymerization, which is the main process that drives cell motility, protrusion of the membrane, which is required for migration. So, okay, so in this, enough talking here, here's the movie. Just kind of take a look at it. I should mention that this is pseudo-colored, so warm colors indicate higher intensity, cool colors indicate lower intensity, right? So I think you can make a few qualitative observations. One is you can see that the cell is moving, right? It's migrating. As it's migrating, this target PIP3 is apparently dynamic, right? It's increasing and decreasing in time. And there's a spatial pattern to it, right? And maybe you can get a sense that the spatial pattern is related to how the cell is moving. So you could make that hypothesis, right? So all this is going on, and we, for many years, have tried to model this process and look at the function of this pathway in cell migration, specifically in fibroblasts, which are mesenchymal cells found in your skin and other connective tissue. Fibroblast migration is very important in wound healing and also in development. I'll touch it a little bit. Okay, and so some of our early work in this area was done by a former graduate student, Ian Schneider, who's now faculty at Iowa State. And so we were modeling this and using sort of some very specific simplifications in order to get a model that we could work with computationally and fit parameters to, right? So we had to make assumptions such as, okay, well, our PI3 kinase enzyme, when it's in the cytoplasm, it's diffusing very fast. Same for our biosensor AKT-PH, it's moving very fast. So we take the cytoplasm as basically spatially homogeneous, right? We on the other hand, we would say that things like the receptor that's being stimulated to recruit PI3 kinase, that it's moving very slow, right? Because it's in the membrane, it's transmembrane. So we're just going to neglect its diffusion and only consider the diffusion of the lipid PIP3, whether it's in complex with the biosensor or not. And then we also assumed equilibrium of the binding between the biosensor and the lipid, right? And we had justifications for all of these assumptions, but we're constantly being asked by reviewers, just do the calculation already. So we say, okay, well, we need to actually solve these a more complicated set of partial differential equations. How are we going to do that? Okay, well, we can break out our Fortran or C or whatever programming language we want to use and code up a numerical solution to those partial differential equations. Not easy to do, right? So it would take several months to do that for a model that's has any complexity at all. And then what happens if you need to change the model, right? Or you're testing sort of the accuracy of the numerical implementation, right? So it kind of became sort of a too hard of a problem for us. So I remembered VCell, and in the summer of 2005, I went to Farmington and I took the summer course and learned how to use VCell. And there was all these things that we hadn't thought about. So the AKT-PH domain, for example, passively goes between the cytosol and the nucleus, right? Because it's a fairly small fusion protein, so it shuttles in and out passively. And there's a time scale associated with that, right? And so even though it's not really doing anything in the nucleus, as far as we know, right, there's still a pool of our biosensor that's in the nucleus. And as the lipids at the membrane sort of recruit the biosensor, there's sort of a redistribution of the biosensor between the cytosol and the nucleus. And we hadn't thought about that at all. So very informative. And then there were other advantages to using VCell. So one is that were important to us. One was making our models publicly available, right? And so not having to worry about providing that code and having it understandable and usable by others was a big deal to us. You know, model reproducibility, very important topic these days. I mean, it's always been important, but it's been more, I guess, highlighted in recent years. And then the final thing was that I wanted to use VCell for teaching. So I had developed a course. I think the original title was Molecular Cell Engineering, and then it sort of morphed into Biological Dynamics. And sometimes I've taught it, sometimes I've taught it with somebody else. And we decided that VCell was a great tool because, you know, very quickly students could learn the software, at least to do basic things, right? And they, you know, no longer had to sort of derive things analytically, and that you could look at very nonlinear systems, which are sort of the norm, not the exception. Okay. All right, so getting a little bit into what we've been doing with VCell recently, I have to talk a little bit more about cell migration. So we started studying PI3 kinase signaling and its link to actin polymerization, because during wound healing, these fibroblast cells are stimulated to invade the wound, predominantly through gradients of spatial cues in their environment. And so this is just showing some of the different types of cues. Probably the one that most people think about are chemotactic cues, right? So this would be a gradient of a soluble chemical, which we call a chemoattractant, that will bind to a receptor on the cell, and the cell will sense that gradient and polarize or at least bias its migration up the gradient, right? So that's what's called chemoattraction or chemotaxis. But the cells can basically interact with other cues. So thinking about wound healing, the cells stimulated or attracted by molecules like PDGF, platelet-derived growth factor. And that's exactly what we were modeling in the context of the PI3 kinase signal, was signaling stimulated by PDGF, whether uniform or presented as a gradient. And then the cell is also responding to characteristics of the extracellular matrix. So the extracellular matrix naturally contains gradients, or at least during wound healing, they naturally have gradients of composition and also gradients of mechanical stiffness. And it's been shown, at least in vitro, that cells will respond to gradients of those spatial cues. And then very similar to wound invasion, where cells are sort of migrating into the provisional matrix of the clotted wound, right? You can also think about cells invading out from a primary tumor, right? And so conceptually, it's a similar process. Okay, and where does PI3 kinase fit into this? Well, I said that PI3 kinase is sort of involved in actin polymerization. Actin polymerization is required for generating the force on the plasma membrane that pushes it forward, right? So it's required for protrusion, which is one of the, I guess you can think of as a key step in cell movement. And so the idea was that, well, okay, this spatial sensing, right, all it has to do is then polarize this signaling through PI3 kinase, and then you'll get more actin polymerization in one part of the cell, and that will control cell protrusion. So that, 15 years ago, that was sort of everyone's conceptual model of chemotaxis. And so you can look at the different ways in which PI3 kinase is activated and what its effectors are. And it all seemed pretty clear, right? So we started getting into this and looking at what is PI3 kinase actually doing, right? And what we found was that when we look closely at movies is that typically what happens is the cell is protruding, and then in order to change its direction, what it does is sort of puts out a new protrusion, sort of branching out from an existing protrusion, at least most of the time. So you would initiate a new branch, right? And this might or might not be stable. In fact, what ends up happening is that the PI3 kinase signaling gets turned on. That's kind of depicted in red in this cartoon. And then that stabilizes the new protrusion. And then the cell will make a decision, right? Okay, which one of these protrusions is going to be successful after you branch, right? And so if these branches, protrusions sort of keep going, right, then the cell will end up executing a 90-degree turn, right? But one of these could sort of win at any point along the way and affect then the turning behavior. Well, what we found was that, okay, that this was important for the cell turning, but the activation of PI3 kinase had nothing to do with this branching process. It only affected the stability. And many other researchers, including us, has shown that if you block the PI3 kinase signaling completely, the cells can still chemotax towards a PDGF gradient. Okay, so we looked at this in more detail, and we found that these new protrusions were initiated through these structures called filopodia. Right? So the cell would put down these little finger-like structures. They're about 100 nanometers in diameter, right? They adhere to the surface, and then those seed the formation of a new protrusion, right? So this is the work of Heath Johnson, who just started his own lab at Hong Kong University. So going international. And so, yeah, so what he found was that, okay, there's this adhesion-based signaling that will cause PI3 kinase to become activated, and presumably or the evidence supported a sort of a positive feedback between sort of signaling, adhesion, protrusion. If you interrupt any of those things, it sort of the system breaks down. So we thought we had this figured out, but we still couldn't figure out what is okay. PI3 kinase is involved in turning. Somebody doesn't have themselves muted again. So, all right. So along came Jim Baer. So he and I have collaborated for many years now. He's a cell biologist at UNC Chapel Hill, who is an expert on the actin cytoskeleton. And what they showed in this Cell paper published 10 years ago is that ARP23 complex, which is involved in this actin polymerization that I talked about, linked up with PI3 kinase signaling, is not required for chemotaxis, right? So this whole mechanism for how you might bias the cell migration was out the window. And so we started to consider this other pathway. And then we later implicated that pathway as another way of regulating the forces in the cell, right? So if you're not regulating the protrusion side of it, maybe you're regulating some other aspect. And what was implicated was the myosin contractility, right? So myosin is a motor protein that interacts with actin filaments and is sort of thought to sort of rein in protrusion by exerting forces on the actin network, right? So, okay, so we had already sort of ruled out this one. What about this one? So here it was required for chemotaxis, And moreover, if you looked at an intermediate of the pathway, namely diacylglycerol or DAG, right? If we image that using a biosensor, it was strongly polarized up gradient, very strongly polarized. So we wanted to explain that with modeling, right? So we turned to VCell. We generated a model in VCell where we have recruitment of this enzyme phospholipase C, which Les's group has done a lot of work on in other contexts. And so when PLC is recruited, it hydrolyzes its substrate, PIP2. This is a lipid, creates the aforementioned DAG. Diacylglycerol recruits protein kinase C to the membrane. And its activation is basically our readout, right? We're looking at PKC activity as sort of the readout of the pathway. And then another important player here was a protein called MARCKS. And MARCKS is a very abundant protein that interacts with PIP2 and sort of buffers it, right? It forms a complex with 3 or 4 PIP2 molecules at a time, and doesn't allow it to be acted upon by PLC. And what we found was that this was a good way of amplifying a gradient. So you have some gradient of the stimulus outside the cell, and this will then be amplified because you're sort of protecting the substrate PIP2 more in the back of the cell than in the front of the cell, right? So even though the total amount of PIP2 is going to be lower at the front because there's more of this PLC enzyme recruited there, the total PIP2 is reduced, but the amount of free PIP2, the available PIP2, actually increases, right? And so this is the work of a postdoc, Kritika Mohan. And then we improved that model through the work of Jamie Nusbish, who also made that trek to Farmington to learn virtual cell, by adding a couple of feedbacks, positive feedback loops that were implicated in the literature, involving phospholipase D and this other lipid intermediate, phosphatidic acid, right? Phosphatidic acid can also interact with PLC, and we hypothesized that then that would stabilize the recruitment of the enzyme. And so we were interested in these properties of this model. How sensitive is it to the gradient? Can a cell, will it polarize, for example, in response to a gradient of only a few % across the dimension of the cell? The answer is yes. We were interested in dose response robustness, okay? Over a range of concentrations of the stimulus, right? Will you still get a nice polarization, or do you really have to dial it in, right? How reversible is it? If I change the orientation of the gradient, if I flip it, let's say, will the system also flip? The answer is sometimes. And then we're also looking at the MARCKS dependence here. So here, just to orient what these curves look like, the red is at the front, blue is at the back, right? And so when you see a difference, that indicates polarization. So that's what we were characterizing mainly in this model. And there were some predictions. So there was, sort of this, if you look at the enzymes that are involved in phosphatidic acid production, right? The main one is diacylglycerol kinase. So you take that product, diacylglycerol, you phosphorylate it to produce phosphatic acid. You might think, well, increasing the activity of that enzyme is just going to reduce diacylglycerol. Therefore, you're going to get less activity, right? And while that's true, right, what happens is that the polarization of the activity gets better. It's improved. And that's just showing here. You can improve by increasing the expression level of this enzyme. You can improve the polarization. If you knock it down, you wreck polarization almost completely. So there were some definite molecular predictions that came out of that. And then we can look at how these different feedback loops work together. We could get the polarization to polarize even more. Okay, and I'm running out of time, so I'm just going to explain one thing and then I'll be done. I have some more stuff I was planning to do, but I'm not going to abuse my privilege speaking to you today. So one of the things we were doing is looking at the structure of the PLC enzyme and how that relates to its regulation and activation, right? So, and this is also important in the context of cancer, because a lot of the mutations found in the structure of the protein are found in cancer, right? And so we looked at, from the insights of papers like this, John Sundek is also an investigator at UNC Chapel Hill. We've collaborated with for a few years now. So Jamie put together this model that accounts for the different states of this protein, all right? And that, normally when you model signal transduction, a very common thing would be to have two states of the protein, active and inactive, right? But obviously it's more complicated than that, right? And so by looking at the structural details of how PLC gamma is regulated, we came up with a model that has 16 states of PLC. We were using the BioNetGen functionality within VCell to do this. This isn't a very complicated network actually for BioNetGen. It only generates 53 reactions, for example, which may sound like a lot, but it's really not. And yeah, and so we study this. And so I'm not going to go through the details of this. This is published in JBC last year. I'll just end there